and where we are going to be all too soon. Okay, so let's see. On Canvas, you should have seen that there was a new posting in terms of an announcement, just kind of laying out what we're going to be moving into coming up. So once we get done with cells, we're going to jump into, at least in lecture, up to the tissue or up to the organ system level. We're going to get to our first organ system, which is integumentary system. That being said, for lab, what's going to happen is there's a little bit more of a transition. So when we were looking at the body overall and its organization, we said that we've got the chemical level, which is like the atoms and then the molecules and things like that. Then we had organelles. Then we had cells. Then we had tissues. Then we had organs. Then we had organ systems. And then we had the organism overall, right? So we're kind of working our way up to get to organ system level. Now for lecture, like I said, we'll go from the cell chapter into integumentary system or skin. In lab this week, you're going to do a little intermediate step. What you're going to do is you will do a lab on tissues. So tissues are going to be where we have a bunch of cells that are working together and structurally usually tied together in some way, and they perform a particular function. And there's 4 basic tissue types that we have. Let me give you a blank page there so we can have those out. So we've got epithelium, we've got connective tissue, we've got muscle tissue, and we've got nervous tissue, okay? So all of the different cells that you would think of in your body are going to fall into one of these 4 categories of tissue types. These are very general tissue types. I think in the announcement I go through them a little bit and talk about the 4 of them. But when we go up a level to organs, an organ is going to be made up of more than one tissue type working together, connected together for a particular function. So if you think about an organ like the heart, right, the heart's going to be mostly muscle because it contracts, but it's also going to have connective tissue, which is kind of holding some of the things together. It's going to have epithelial tissue, which epithelial tissue is kind of a covering and lining. So lining the inside of the chambers is going to be epithelial tissue. Lining the outside of the heart is going to be epithelial tissue. So it gives us an example of an organ where there's lots of different types of tissues working together. Nervous tissue even, there's some in the heart too, to get it to coordinate, to speed up, to slow down as we need. Okay? So in lab, what you're going to do is you will, the lab exercises that you're doing is one on tissues and one on integumentary system. So the one on tissues kind of goes through that in a general way, and then it's going to go through epithelial tissue a little bit more in detail. You're not going to go through all the 4 tissue types, but just focus on epithelial to give you an example of the fact that there are a lot of specific tissue types that exist in the body too. So for instance, of the epithelial types, there is what's called simple squamous epithelium, which is like only a single flattened cell layer thick. When we need a thin layer where we can have things maybe diffuse through very easily, like across the capillaries in our cardiovascular system or across the respiratory membrane in our lungs so we can have gases exchange. We want a lining, but we want a very thin lining so things can get across easily. There's also what's called stratified squamous epithelium. So stratified means that it has layers. Squamous means that they're flat. And your skin, or at least the epidermis of your skin, is a good example of stratified squamous epithelium because it's got lots of layers, which gives us a lot of layers of protection instead. So each one's a little bit different. Cell shape's different. Number of layers is different depending on the function. So it's a good example of this overlap between form and function. Okay? Now, if you were doing it in the classroom, in the lab, you'd actually pull out the microscope that you learned about last time, and you'd use that microscope to view the different epithelial types and see the differences in the number of cell layers and shapes, etc. You guys will use some of our online resources where we've got these web albums of all the different slides, of all the different models and stuff like that, where you will go and view the different tissue types that the lab manual is asking you to view, but you'll just view it online. So I gave you the link in the announcement, and I updated the directions in the module, and it gives you the link of where you need to go to find all those slides. and they'll have all the epithelial slides there. They're all labeled with their names and stuff like that. So you can just use that to view your microscope slides instead of what it's telling you to do is bring out the microscope and do it that way. Okay? The second lab is integumentary system, and that will go over much like we're talking about in lecture coming up. So that'll have a good overlap. But like we said, tissues, the only place that we are going to address tissues is we'll kind of take a small little jaunt into tissues in lab, but we won't go through chapter 4 here in lecture. Chapter 4 is tissues in the book. You can certainly read through it. You can use your book to see different examples of it to help you with the lab and stuff like that too, but we won't do a whole lecture on tissues. Likewise, let's see, so I think maybe chapter 5 is tissues. Chapter 4, I think, is metabolism, cell metabolism. We're in the middle of the cell lecture right now, and what we're going to do is we're going to kind of bring chapter 3 and chapter 4 together. Right now we're talking about different organelles inside of the cell. Towards the end of the chapter, what we're going to do is we'll talk a little bit about cell metabolism, but we'll just kind of co-opt it into this lecture instead. So that means you don't have to read chapter 4 in the textbook either. You can if you want to, get some more information about it or think about, what we talked about in terms of producing ATP, et cetera, but you don't have to. We're kind of co-opting it into what we're doing now. Okay? Okay. So, yes.
Why is there so many different labs?
So many different labs. So that's a good question. So when we look at the directions for this module, like we said, Canvas tries to be all holistic, where it's talking about a particular topic. Usually it's a system, like cardiovascular system, and then it says this is lecture and this is lab. Well, for cells and tissues and stuff like that, like I said, we don't really do it in lecture. We kind of do it in lab. So what we do there is we split it into 2 weeks of labs. First, you're doing microscope and cell and learning how to do that. That was last week. Next one you're going to do is you're going to step up to tissues and work into skin. And so since they're all kind of related as we're looking at organization of the body, we're kind of co-opting them into that same module. So it's just I didn't create a separate module because we don't do a lecture on tissues and stuff like that. So it would be kind of weird if we just had a module on tissue and there's no lecture in just lab. So that's all. Yeah. So usually it'll just be the one lecture and one lab, and it'll be the same each week. Occasionally, if it's a complex system, we may have more than one lab associated with it. Just like sometimes we have more than 1 week of lecture that will be dedicated to a particular system. Okay. Other things before we move into cells again? Okay. So the last time, what we did is we were finishing up talking about some of the organelles. We had talked about those that help to produce proteins. We talked about the role of the nucleus as a cookbook and things like that. Then we talked about the organelles that help to produce proteins, the rough endoplasmic reticulum, smooth endoplasmic reticulum, and Golgi complex, modifying, packaging, exporting proteins, etc. So now what we're going to do is we're going to finish up the tour of the different organelles in the body. Yes, sir?
So is that what the ER stands for?
Yes. Endoplasmic reticulum. So if we look at it like there, that's where we can see it there, yeah. And it brings up a good point. So like usual, we're learning the terminology, so it is important that you usually spell these things out. If you write it out the first time and we're talking about it later on, and I know you know what ER means, and it doesn't mean emergency room, then you're fine. But for the most part, you should kind of show your knowledge, like if we're in the essay section of the exam, spell the things out so that I know you know what you're talking about. Okay. Yeah, so Jessica was asking what the question was, whether ER means endoplasmic reticulum, and it does. Bless you. Okay, so looking at other organelles, we've got the mitochondria, and this is one of the organelles that people tend to remember the most from their, biology in high school or something. These little bean-shaped organs here are the ones that produce ATP in the cell. Now, they're the major site of production of ATP in the cell, but it's not the only site. We'll learn at the very end when we're talking our little introduction to metabolism that there are multiple steps to producing ATP in the cell. The first one occurs out in the cytosol. The other two occur inside of these little ATP factories. And the ATP, anybody remember what ATP stands for? So it is adenosine triphosphate. Jordan was all over that one. He typed faster than I did. So you can see ATP there. Okay? So adenosine triphosphate is kind of like a molecule where, much like a rechargeable battery in your phone or something like that, right? You expend a bit of energy to charge it up, and then you can take that energy with you where you need to go, right? You have a rechargeable battery. You can expend that energy later. So the energy that we take in, like when we're eating and things like that, right, those nutrients, we are taking them in a certain form, like glucose, etc. Then what happens is your cells use that glucose to manufacture ATP. We store the ATP, and then that can be broken down and used, again, like a rechargeable battery, later on if the cell needs it for moving, if it's a, if it's a muscle cell or something like that. Okay? So the mitochondria are the organelles inside the cell that produce the bulk of the ATP that these cells need. Okay, so once we move forward, we'll find that there's a couple other organelles that are fairly simple in terms of their structure. The mitochondria, when you looked at that, it had two membranes on it, and has all these folds or cristae in it and stuff like that. It's fairly complex in terms of its structure. For the next two, these are fairly simple. They're kind of bags of enzymes, okay? So one of these organelles is called a lysosome, and the other one is called the peroxisome. And they both look like little bags, and you see them on the cell model, like you use for the quiz and stuff like that. It's like one's a yellow dot and one's a red dot. They're really indistinct in terms of their anatomy. But the chemicals that they contain within them is what sets them apart. So lysozymes use enzymes. Remember, enzymes help chemical reactions go in order to destroy foreign substances and cellular debris. So with lysozymes, they can, if it's a particular cell, like a white blood cell, it can work to get rid of a bacteria it's engulfed or something like that. Or if there are parts of the cell that are worn out, like parts of the cell membrane or something, they can take in those parts, digest them, if you will, and then kind of recycle the components. So in a way, it's kind of like the digestive system or something, or the immune system of the individual cell. The peroxisomes, like we see here, house enzymes as well, but these are oxidative enzymes. And so these, in a way, I guess if the other ones are more like the immune system, like the lysosomes, the peroxisomes may be more like the digestive system. Again, the digestive system is not really a waste or excretory system, but ultimately it has ways of trying to help get rid of waste products. So Jordan was asking, do enzymes destroy old organelles in the cell? It certainly is possible if an organelle wears out or something like that, or a part of it does, like the membranes of ER or something. It's possible that it could be recycling that way. But with those, it's very important that with the peroxisomes and the lysosomes that the enzymes stay within the particular membranous sac, because it's like almost like the stomach and stomach acid and enzymes there. If you release it onto something, it's going to digest whatever it gets onto. So you want to make sure that it's contained. So if there's something you need to recycle or get rid of, you want to put it inside of the organelle, and then it digests it that way. But certainly possible to recycle various parts of the cell. Okay, now as we continue on talking about things that we find inside the cell, if we remember, we're making these links between the cell as a living unit and us as a living unit. And so for us, we have different organs that support us. The cell has different organelles. We have a skeleton that supports us, allows us to move and things like that. It also has a skeleton. Now, this is called a cytoskeleton. Cyto means cell, right? And so it's going to have a structure to it, but it's not going to be bone like our skeleton. It's actually going to be made up of proteins. So now we're seeing another function for proteins in the cell, right? So we have, the ER and the Golgi complex and stuff like that making proteins. Here's a whole other set of proteins that we need in order to give the cell structure. So there are three different types of cytoskeletal components. One of them are called microtubules, because you can see they're kind of tubular, they're kind of open if you look at the end. Another set are called microfilaments, and the last set are called intermediate filaments, because if you look at the size of this one, it's in between the microfilaments and the microtubules. So each one is a little bit different in terms of its structure, and again we keep that link between structure and function, so they may have different functions accordingly. So with the microtubules, right, these are kind of the hollow ones. Some of their functions can be transporting secretory vesicles. So these are going to be ones where maybe we've made proteins and we want to move them out of the cell. Movement of special cell projections such as cilia or flagella. So cilia are going to be small little hair-like projections that can be on the surface of a cell. Now if you were talking about a cell, like if you're looking at a sample from like pond water or something like that, there are unicellular organisms. Organisms that are just made of one cell, they use cilia to kind of move themselves around. They'll swim that way by beating these little hair projections. For our cells, since our cells are usually connected to each other, they use their cilia for moving things past the cell instead. So for instance, along our respiratory system, we've got mucus lining it, and that mucus, one of the jobs of that mucus is to catch things like dust particles, etc., that we may inhale. And then we don't want them getting down to the respiratory membrane. We want them out. So it gets caught like flypaper in the mucus, and then the cilia kind of beat like this and bring it up, 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 up out of the respiratory passages, kind of over a hill of the pharynx, and it actually goes down into the esophagus and down into the stomach instead. Yeah. So when you see those people and they're like, right? They're just aiding the process there. They're just kind of hurrying it along. It's the natural path of things. But they actually, the term that they use for that is actually mucus escalator, because it has the mucus on top of the cells and those cilia beat it so that it goes up away from the delicate respiratory membranes. Okay, so those cilia and movement of them are according to microtubules. They help with that. And distribution of chromosomes during cell division. So we learned about mitosis last time, right? We're duplicating the chromosomes, then we have to move them to the poles. So when you see those spindle fibers and things like that, those are made up of microtubules. So it's helping to move those different chromosomes to where they need to go. Okay, so then we have microfilaments. These are not hollow, right? So these are the smallest ones. The microfilaments work in various contractile systems. When we get to the muscular system, we'll learn that your muscles shorten through what's called a sliding filament mechanism. You have protein filaments in there that slide past one another to allow those muscle cells to shorten. And so those are made up of certain types of microfilaments. And then mechanical stiffeners and cellular projections. We'll learn that in certain cells, like in the digestive system, we need a lot of surface area, because anywhere along that cell is where we can absorb things across the wall and get those nutrients inside of us. And if it's just flat, well, then we have that surface area. But if it waves back and forth and back and forth and back and forth like that, there's a lot more areas for things to get into the body. So we have these little finger-like projections like this on top of the cell that gives us all this surface area to be able to absorb things. And these, they're not hairs. They're just, the cell membrane, instead of being flat, goes back and forth and back and forth. And so these are called microtubules. If you're going to get benefits from microtubules, they can't be laid over on each other like this, because now there's no surface area again. They have to be sticking up like that. And so the microfilaments help these little microvilli to stick up. Then we also, there are some microfilaments that are involved with movement. Interestingly enough, there are, like in the blood, the white blood cells, there are certain type of white blood cells that can actually move on their own. Most people think about the blood cells just kind of being pushed around by the plasma, going through the arteries and veins and stuff like that. But the white blood cells work to protect you. They want to get to the site of infection. Most of the time if you have a cut, right, your infection is not inside of the blood vessel. It's like in the skin or somewhere like that. The white blood cells, if they're going to protect you, they've got to get to the site of infection. So white blood cells, certain white blood cells will actually migrate out of the blood vessels using this kind of what they call a amoeboid movement. They'll actually, like, have you ever seen like a snail move? Like if you go downstairs and you view the saltwater tank, you see the snails on the side, they extend out their foot like this, and then they kind of pull themselves along. They kind of change shape that way. the white blood cells, using microfilaments, will do that type of movement. And they'll squeeze out of the blood vessels and then migrate to the site of infection so that they can kind of enlist in the battle that way. Okay, and then the last one, we don't have a particular slide on, but the last one was an intermediate filament. And intermediate filaments usually give cells a certain amount of strength. So like in the skin, we'll learn that there's a number of layers of the epidermis that have a lot of intermediate filaments or keratin within them. And you hear about the hair and stuff like that, right? So it gives those cells strength, extra strength for protection or structure or something like that. Okay, so all three of those are different examples of proteins that help to make up our cytoskeleton. All right, so now what we're doing is we're getting our way back. We've gone through a lot of the organelles. Now we're kind of getting our way back to that very first thing that we covered in terms of the nucleus and its role in terms of maintaining the cell and creating new cells. And we talked a bit about this already because we jumped forward to talk about mitosis last time, so you were prepared for the labs last weekend. But this is a place where we can see it again. So with the nucleus, we said that it's like a cookbook, right? The nucleus contains both DNA, that's deoxyribonucleic acid, as well as other types of specialized proteins, like different types of transport proteins and stuff like that inside. It's got two different roles. Number one, it's a cookbook so that we know how to synthesize proteins. And number two, it's a blueprint for cell replication so that we have an epithelial cell. When it divides, it makes another epithelial cell, not a muscle cell, stuff like that. Okay? It is fluid-filled so that we can move our different strands of DNA around. It has a nucleolus, which is almost a nucleus within the nucleus, that has basically RNA and protein in it. And then usually, if the cell's not dividing, it's got the DNA in this form called chromatin. We said, like last time, when we were talking about mitosis, we said that when we're preparing for mitosis, like in prophase, we're winding up the DNA into chromosomes so we can move them very easily. If we're not in a dividing mode, then the DNA is kind of unwound so that we can read it, right, so that we can make the different proteins and stuff like that. So the chromatin is kind of found in the outside of the DNA. Outside of the nucleolus, but still in the nucleus there. So this is kind of a graphic showing the process like we were talking about in terms of the creation of proteins. So we can see here's the DNA inside of the nucleus, right? We split it apart so that we can read a certain gene, right? So there's going to be an area where we start reading it and an area where we stop reading it, right? So just like a cookbook, if you're reading a recipe, it's page 7 to page 10 or something like that, and that's it. So then it's going to be read, and this is going to be read with the help of the messenger RNA, right? So it's going to read the particular gene. It's going to go out of the nucleus, because it's got to get to where the ribosomes are, because we said the ribosomes are the factory for where we produce the proteins. So here, this little brown ball that you see here, that's the ribosome. It's made up of, in essence, ribosomal DNA, or RNA. So it is the site of production. And then you see these little things here kind of flying through, those little crosses that you see here. These represent the transfer RNA, or tRNA. So tRNA, once the ribosome is kind of reading the messenger RNA, the tRNA are kind of like the cook's helpers, like we said. They're grabbing the individual amino acids, like you see here in color, and they're bringing it to the ribosome so that we can assemble that polypeptide chain, link them together that way. And every 3 different nucleotides in that gene, like you see here, is a particular code for a particular amino acid. Okay? So AUG is like a start code, and it also, in essence, codes for the amino acid called methionine. So that's the recipe. You read those 3 at a time and says, okay, I need this one, I need alanine, I need threonine or glycine or whatever. Okay? So that's the recipe. It reads those 3 at a time. It tells the transfer RNA to bring those in, and then it links them together to make a polypeptide and then ultimately a protein. Okay? And that's happening all the time, whether it's proteins that are needed inside of the cell or whether the cell is making these for export, like hormones or anything like that, or enzymes, etc. Now, one little side note before we kind of finish up the last couple topics of this chapter. A lot of the times when we look at different books and we talk about anatomy and physiology, you think, wow, everything that they know about the body, they've already discovered, right? It's like, it's not like we've developed a 207th bone or anything like that. So how come we have all these different textbooks and new textbooks and stuff like that? Well, there are still developments that they are discovering through time. This one right here is an organelle that they don't necessarily know what it does. Right now, what they've called it is a vault, because they think that it's made up of two components that come together to make a barrel-shaped structure. And so what they think is, they think that maybe it's there to help transfer the parts of the ribosome to the place where the ribosomes themselves are connected, or maybe it transfers the messenger RNA out the different pores in the nucleus out to where the ribosome is. But they're not sure. They can see them using an electron microscope, and they can see they have this octagonal shape. It's not just round. It's like a barrel like that, but they don't exactly know what it is. So that's kind of an active area of research that they're still trying to figure out, even in a regular generic individual cell. So not everything about anatomy and physiology is known under the sun yet. Okay, so that takes us through our tour of the different organelles inside the cell. The other thing that we hit in terms of a topic related to the cell is membrane transport, because when we look back at the different functions of a living organism, we talked about how it needs to respond to the environment, how it needs to get nutrients and oxygen, it needs to get rid of waste products, etc. So the cell membrane, we said, is kind of like the border or even like a door or something like that, right? It's not solid to where it prevents any movement. It just regulates the movement so that we can make sure we know what's coming in and what's going out. So we need to understand the different ways that things can come in and come out. And so these are different types of what's called membrane transport. And you've likely heard of these before, so hopefully this will just be a refresher. Now, for the membrane transport types, we find that we can separate them into two different categories. So this first example here we're seeing is a type of unassisted transport. It's also one that could be called passive transport. Okay? So passive transport, if you think about the difference between active and passive, right? So if you're in a group and you're deciding what movie to see or something like that, right? So if you're in the group and you're just passive about it, then whatever the group decides, that's what you'll do, right? You don't really care. You kind of go with the flow. If you're active about it, then you're expending energy to say, no, I don't want to see that. I want to see this, right? You have to expend energy to do so. Transport across the plasma membrane can be that. We can categorize it in that way too. We can expend energy to actually like push something across, or we can just let it kind of go the natural way it would according to like different gradients, like going from high concentration to low concentration. The other category of transport is whether, when we get across the membrane, does that particular ion or molecule need assistance? Does it need to go through a channel or something like that? Or can it just slip right through the plasma membrane with no problem whatsoever? Okay? So our first type of membrane transport that we are looking at is called diffusion. And diffusion is based on the random movement of molecules. So these molecules, that are smaller than what we can see in the microscope, kind of have a little bit of energy to them. It's called Brownian motion. They kind of vibrate, if you will. And when they are packed closely together, they tend to bump into each other more often because they're packed tightly together. When there are fewer of them for a given space or volume, then they don't bump into each other as much. It's kind of like people, right? So the molecules like their own elbow space. And if they don't have that, then when they're packed in tightly, they're kind of looking for an area that's not quite so crowded where they can get that elbow room. So what we find is that for diffusion, in most cases, it centers around the concentration of a particular molecule or ion. Okay? So like we said, these molecules are looking to go from an area of high concentration to low concentration so they can get that, if you will, molecular elbow room. So it's kind of like the typical example I use for that is say you were at a party or something like that, and it's say it's like a 2-bedroom house. So you're in a room, and everybody's there, and you've got your drink, your non-alcoholic drink, of course, and you're, just feeling a little crowded. People are bumping into you, and you're spilling your root beer on yourself, and you look into the other room, and there's much fewer people in there, right? And they're sitting on couches, and they're just relaxing, and you're like, oh, I'm going to go in there because this is too crowded in here. And you'll naturally move from an area of high concentration to low concentration, right? Now, that will likely continue. People will go, oh, yeah, that is much better in there. And that movement will likely continue until such point when somebody looks in the other and they're like, all right, I'm feeling crowded in here, but when I look over there, it doesn't look any better. So why should I bother going over there? So it'll reach what's called equilibrium, where it's basically the same concentration on either side of the membrane. So the molecules are like, what's the point? You might still get a random person that go, I'll try it out over there. But there's not this mass exodus going one way or the other. So it's the same thing for the molecules. So diffusion, when there's a big gradient, like it's really high concentration here and really low concentration here, it'll move, a lot will move from high to low. It's like a ball rolling down the hill. Steeper the hill, faster it moves. Little hill, yeah, it'll go, and you don't have to push it, but it won't go as fast. Okay? So that is basically diffusion. And so that's very important for lots of things in the body, like oxygen and carbon dioxide exchange. That occurs from diffusion. You take a big breath in, you have a high concentration of oxygen in your lungs. The blood that's returning to the lungs has dropped off a lot of oxygen to the tissues, and so it has a relatively low concentration. So the oxygen naturally diffuses from the lung into the capillaries from high to low, and that's how you pick up that extra oxygen that then gets transported around. Okay? Okay, so for diffusion, usually what we have here is an unassisted type of diffusion that is going to move from high concentration to low concentration until it reaches equilibrium. Okay? And like we said, there are various examples in the body of when diffusion is very important, and we'll hit those along the way when they become relevant for a particular system and our understanding of it. Now, osmosis is something a little bit different. Okay? Osmosis is a very specific type of diffusion. It's when water itself is diffusing down its own gradient. So it's where you have a high concentration of water and a low concentration of water, and the water molecules are diffusing from high to low. But you got to think about this one a little bit more. Okay? So with this one, what we have here is, say this is a beaker, okay, like this, okay, and it's got a membrane, a permeable membrane in the middle of it, which is like the plasma membrane, okay. The water molecules here are in light blue. The solutes are in purple. Now, a solute, by definition, is something that is dissolved within a fluid, in a solvent. So if you're making Crystal Light lemonade, okay, water is the solvent. That's what is the liquid, and the solute are the crystals of the Crystal Light that you're trying to get to diffuse in there, okay? So what happens is, when we look at a solution, if it's 100% water, well, that's the highest percentage of water you can have. That's a high concentration of water. It's 100%. If you add anything to the water, now it's different. Now it's like 90% water, 10% Crystal Light, right? So now what's happened is, for that solution, you've got a lower percentage of water because you have the solute in there, right? So it's like 90, 95% or something like that. So at that point, when you have two solutions that have different concentrations of water, the water will actually move from an area of high concentration of water to an area of low concentration of water. So osmosis is that. It's the diffusion of water itself. But in order to get a concentration difference of water, you got to throw some stuff in with the water so that it's like 50% water and 50% solute, 90% water or something like that, and 10% solute. Okay? So it has to be mixed. So that's what we see here, is that on the left side, we see a higher concentration of water and a lower concentration of whatever else, Crystal Light or purple Kool-Aid or something like that. On the right side here, we see more solute and less water molecules. So there's a natural gradient for water to diffuse this way through osmosis. And it will do so until it reaches equilibrium. Okay? Okay. So that's very important because we want to be in balance in our cells in terms of the amount of water they have in there. Cytosol is mostly water. Outside of the cells, we have water. We want to make sure that it's in balance. If we get a lot of movement of fluid into and out of the cell, it actually can change the volume of the cell, and it can swell up, and it can shrink accordingly. So that's when we come to this thing that's called tonicity. So tonicity is the effect of a solution and its concentration on the cell volume. So what that means is, like we said, if you have a lot of fluid moving into or out of the cell, like a balloon with air, right, it's going to get bigger if you're packing things into it, or it's going to get smaller. And that can affect the cell's performance because, like, if it's a neuron and it's supposed to be relaying muscle signals, but it's swelling up, it's not going to be able to do that very well. So what we find is most of our cells are isotonic, right? Iso means the same. So isotonic means it's the same concentration of the solutes inside the cell as outside. So we don't have a lot of water moving one way or the other. If, on the other hand, it's not in the same concentration, then what's going to happen is we can see changes in cell volume. So what we see here is a red blood cell. Okay? If you put it in a solution that has the same concentration of solutes, they don't have to be the same exact thing, but if we count them up and we see that there's like 300 inside the cell, we need to have 300 of these molecules or of some type of molecule outside of the cell, and then the concentration is the same, water is not going to be flowing much either way. In this case, right, inside of a red blood cell, it's about 300 milliosmoles. That's the concentration of the solutes. If we put a red blood cell in a solution that only has 200 milliosmoles, then this is what's called a hypotonic solution. Hypo means low, right? A hypodermic needle goes under, or is low, underneath the skin, underneath the dermis, okay? So if you put a red blood cell in a very dilute hypotonic solution, now it's a high concentration outside of the cell compared to inside of the cell for water. So the water starts moving into the cell to try to even it out, and the cell swells up. If that concentration difference between inside and outside the cell is too different, that cell will continue to swell until it actually lyses or explodes. So that's pretty important, right? Flip side of the coin can also be bad, right? If you have a very concentrated solution outside, now you've set up the gradient the other way, right? There's less water outside the cell and more solutes. So now the water is looking across the membrane and goes, okay, there's a lot of me here inside the cell and not as much out there. So I'm going to leave the cell until we reach equilibrium. And that's going to cause the cell to shrink. So when you get dehydrated, that's what happens is you don't have enough fluid in your tissues and stuff like that. The cells are actually losing fluid to the surrounding tissues, and they will actually shrink. And that will decrease their performance. If it's a muscle cell, it won't be able to contract well. If it's a neuron, it won't be able to send signals, etc. So it's interesting because sometimes you hear examples of when that goes awry. Your body's always trying to make sure that the concentration is about the same, etc. You hear about those examples of water toxicity as well, right? You drink too much water too fast, the body can't adjust the concentrations of things fast enough and keep it at homeostatic levels. So there's an example here and in California of two cases where the one here was actually a radio show contest. And what they were doing is you had to drink water, I don't know, it was like gallons and gallons of water as quick as you can. And whoever finished it all won, right, Justin Timberlake CD or something like that. Okay? And so, right, they're figuring out, it's water. What's going to happen? It's good for you. It doesn't have anything bad in it. Nothing's going to happen, right? So they go to the contest, and they're chugging the water and whatever. The woman that loses feels a little woozy afterwards, but totally fine. She goes home later, ends up dying from water toxicity. Because what happens is she ended up, in this case here, in the middle. She ingested so much water, she diluted all of her her extracellular fluid, and the body couldn't get rid of it fast enough. So outside of her cells is very dilute. Now there's that old concentration gradient. So all that water starts going into the cells, into the cells, into the cells, and they basically exploded and she died. Yeah. It's also what happens if you take like a saltwater fish and you put it in a freshwater tank. You can't hear the cells explode, but they do. Yeah. You don't want to do it the other way either. Because the other way, right, you take a freshwater fish and you put them in a saltwater tank, then that's what happens. All their cells shrink up because they're losing water the whole time. If you take my marine biology class in the spring, we'll tell you all about that, how freshwater fish and saltwater fish actually deal with that all the time. Okay. So we've talked about things like diffusion and osmosis, and those were usually types of unassisted transport. These molecules will go into and out of the cell without such a problem. Okay, they don't need to be assisted. When we get to assisted transport, usually you have a carrier molecule, and a carrier molecule is going to be like a protein that's embedded in the plasma membrane. It has a complementary shape so that it binds with something else like a molecule in a lock and key mechanism. And when it finds it, it binds to it, and then it folds in and it transports it across the membrane. It escorts it. Okay? So with this type of transport, it can be active or passive. If the molecule itself is going from high concentration to low concentration naturally, then its only issue is it can't get across that lipid bilayer. So maybe it's a hydrophilic molecule and it can't get across that fat hydrophobic core. So it needs to be escorted across. But it's the way that it was going naturally. Okay? So that would be passive transport. It doesn't require ATP to push it. It just needs to be escorted across. Okay? It's like a Cub Scout taking an old lady across the street, right? She can get her way across. He's not having to push her, but she needs to be escorted across. Okay? There is active carrier-mediated transport instead, though. That would be like, no, hey, old lady, you can't come here. You need to go over there. And you're actually physically pushing and making them go across the street that way. Okay? So in the case of molecules, that would be if you have high concentration and low concentration, and you have a molecule that wants to go against the grain, if you will, or against the flow. It's like pushing a ball uphill. In order to get it to go that way, you've got to expend energy to get it to go that way. Same thing in terms of the cell. And because it's carrier-mediated, like we said, it can be specific so that the particular molecule, the protein, matches with the particular molecule that it's transporting. It can become saturated if there's too many things to transport, and there's only so many of the Boy Scouts. Right? If you get a big busload of old people that just got back from Las Vegas and they all need to get across the street at the same time, you got 2 Cub Scouts, it's going to take some time, right? They get saturated. They can only do so much. And then there's competition, too. In terms of old people, right, they're all fighting to get escorted first because they want to get to Old Town Buffet earliest at 3 o'clock, right? But they can all go according to that Cub Scout. There are receptors that can bind multiple molecules, but if there's a lot of them, then there can be competition for transport that way. Okay. So with assisted types, like we said, we can have passive. So there's like facilitated diffusion. That's like we said, it's going the way it would naturally go, but it needs an escort. So that's called facilitated diffusion. A facilitator makes things easier, right? So this is a transport molecule that is going to escort something across the wall. Glucose, that sugar molecule that we need, that's the way it gets inside of cells. It doesn't diffuse across. It can't get across the lipid bilayer. It needs an escort. But it would naturally go from high to low. We just need to escort it across. So that's the case that we see here, right? See high concentration here, low concentration here. That's the natural gradient for it to go that way. But we need this escort molecule here, the transport molecule, that has a particular binding pocket that fits it right, to bring it across like that. Okay? So that's facilitated transport. Active transport is also assisted, but this, active, means it requires energy. This is where we're pushing molecules against the gradient instead. So if we look at it, we've got to burn energy, burn ATP to do so. This is the example. Here's the high concentration here. Here's a low concentration here. So the gradient is going to be, the natural gradient is to go this way. But what this molecule is doing, this transport molecule, is it's taking these things and it's continually pushing them from low to high against the natural way that they would go. Yeah? Oh, sorry. Yeah, I missed his other one there. Yeah, Jordan, if ever it seems like I'm ramming over there, you can raise your hand like that, and it'll give us more of a indicator. So Jordan was talking about aquaporin channels in cells. So if we look at the aquaporin, that term, it's a particular channel, and aqua makes you think of water. So aquaporin channels are going to hasten the movement of water across the cell membrane. So in the urinary system, it's very important that we very quickly can reabsorb water if we need to. Like if you're dehydrated, you want to reabsorb water and not let it go out the urinary system. And so if you put these extra channels in there, we don't have to wait for it to trickle across the cell membrane. Now we've got big tubes that can let it go across as well. So that's kind of unique to that system. The other one is just a follow-up. It says, couldn't water just diffuse through the osmosis? It's going to hurry up the process because as the solution that we got out of the blood is going through these little tubules in the kidney, we've only got so much time that's passing through before we lose it as urine. So if we need to reclaim it, we got to do so quickly. Yeah. Okay. So active transport, like we said, uses ATP. Okay, uses ATP. There is a difference between what's called primary and secondary active transport. The details about that isn't too critical. I wouldn't worry too much about that. So let's move forward so we can look at other types of transport. You'll also hear in certain systems about what's called vesicular transport. This is where the plasma membrane kind of envelops or creates a vesicle around whatever is being transported and brings it across the membrane like in a big bag. And so there's different types of vesicular transport. If it's coming into the cell, we call it endocytosis, right? Endo means inside the cell. We're bringing it in. If it is going out of the cell, that's called exocytosis, exiting the cell. And so exocytosis is like when we're exporting proteins, we put them in the vesicles in the bag, and they go out of the cell that way, okay? Endocytosis, there's actually a few different types, and a lot of these are done like by white blood cells, et cetera. They need to grab something large and bring it in the cell, not just a molecule at all times. So the differences there are pinocytosis. Pinot makes you think maybe of a Pinot Noir or something like that, right? So that means cell drink, and so in that case, it's taking in some molecules, but it's taking in some surrounding fluid as well. Phagocytosis, phago means to eat, and this is where it's taking in some large molecules or a cell, like a white blood cell, it grabs onto a bacterium and takes it inside to digest it. And then receptor-mediated endocytosis, this could just be for particular special molecules or larger structures that we want to bring inside the cell. It's very specific. It uses receptors to bind to it, to identify it, and then it takes a large chunk of it in as opposed to just one thing. But all of those are different types of vesicular transport coming into the cell. Now, one other type of movement across the membrane that we'll hear about when we get to urinary system again is going to be filtration. And filtration, it's not about concentration gradient like we were talking about before, but it's about fluid pressure. And so filtration, if you think about it, did we talk about coffee before? Now coffee's made, right? So it has a filter in there, right? So you put the filter in there, you put the coffee grounds in there, and then the water comes in from the top and goes through the filter, and small molecules like water and like the small diffused coffee molecules come through the filter, but the big coffee grounds stay in, right? And it's the pressure of the water driving it through the filter that allows that to occur, okay? So in filtration, the same thing can occur where, like in the urinary system, we're using the force of fluid, like plasma, to push these molecules through a size-selective barrier, like a colander or something like that, right? You make pasta, dump that into the sieve or the colander, small things get through, big things stay in the filter. So we will see that as well, that there are specific cells that allow for filtration also. Okay, so this slide here just goes through and lists all the different types of membrane transport that we went through. So you need to know the different examples and what they mean. So like diffusion, what's diffusion, stuff like that. You should know whether it's passive or active, know whether it's assisted or not, things like that. So for instance, you could have like a short answer question on the test where it says, name 2 different types of passive transport and tell me, you know, the difference between them. Or it could be more of a multiple choice question where it actually gives you the definition for osmosis, and then you've got to pick it out in terms of the answers instead. Okay? Okay. So that, after talking about membrane transport, then that gets us up to cell cycle. Now this we already went through because we were trying to prepare for the mitosis lab from last week. So does anybody have any questions on prophase, metaphase, anaphase, telophase, that type of thing? Seems okay? All right. We'll move through a little bit of that. And then it gets us to the last topic of cell, which this is where we're kind of co-opting some of the Chapter 4 information into this lecture about cell. So like I said, we're not going to do a whole metabolism chapter, but metabolism is all the different chemical reactions inside the cell that keep the cell alive, etc. And a lot of those chemical reactions have to do with the production of ATP, okay? Because ATP is going to be the energy molecule that allows a lot of other activities in the cell to occur. So what we're going to look at here are different ways that we can produce, or a cell can produce ATP. Okay, so it's in essence, it's a three-step process, okay? The first step in the process is what's called glycolysis. Now, this is going to be kind of a summary. It's going to be like a Cliff Notes type of situation compared to what you'll see in the chapter. And do you guys not have these slides? No? Yes? No? Is it in your lecture notes, these? They are. Okay, just making sure. Just making sure. I saw lots of people writing sometimes, you know. Some things get a little bit off. Okay, so glycolysis here, it occurs out in the cytosol. And what it does is it starts with one molecule of glucose, which is a simple sugar, and through a series of chemical reactions like we talked about, like synthesis reactions and decomposition reactions, at the end of it, we get two different things. Number one, we get two molecules of pyruvic acid. We've changed the original glucose molecule, and we've made it into pyruvic acid. But in addition to that, we get two ATP as well. Okay? So that's a pretty good return, right? Start out with one glucose, and through these chemical reactions, you get two ATP. All right, so that's pretty good. Now, when we look at this process here of glycolysis, of cranking out two ATP for every one glucose, does not require oxygen. You know, we think about our cells, okay, yeah, as a living thing, we need oxygen, our cells need oxygen, etc. The main thing that our cells need oxygen for is to produce ATP in the mitochondria. This first step that we're looking at here is occurring out in the cytosol, not in the mitochondria, and it doesn't require ATP. So this is what's called anaerobic production of ATP. So anaerobic means without oxygen. A like without, and aerobic is talking about the use of oxygen. So when you, for instance, go to the gym, you're talking about doing aerobic activity, right? You're supposed to do long periods of time, using a lot of oxygen, not exerting yourself too much, but burning calories and stuff like that. If you start to do something that's really intensive, you'll find you can't do it for very long, right? You start sprinting on the treadmill, you can't do it for very long. Because what will happen is your cells will burn through the ATP stores that they have, and that whole process in the mitochondria of producing ATP uses oxygen, and it takes some time. And so you quickly outpace. You're not getting enough oxygen to the cells fast enough. They're not able to manufacture the ATP that way. And so you're left with only this process here of cranking out two ATP for one glucose, and it's quick and dirty, but you don't generate a lot of ATP that way, okay? So this is anaerobic. So you can only rely on this for so long. That's why you can only sprint for so long before your muscles start giving out, because all of a sudden oxygen demand is too high in your cells and stuff like that. They have to rely on this, and even though 2-to-1 seems good, you'll find out that there's a much better ratio when we look at aerobic production instead, okay? So the two other steps of ATP production, the first one is called the citric acid cycle, and the second one is called the electron transport chain. And so Jessica was talking about why bikers blood dope, allegedly. No, I'm just kidding. Yeah, some do. The idea there is, yeah, so oxygen demand, when their muscles are using so much energy, they need to produce ATP. The idea is when they blood dope, they're increasing the number of red blood cells in their blood, That means the red blood cells, their main job is to transport oxygen. That means that we can get more oxygen to the cells faster. That means they can perform longer and stronger. So that's the idea with that, yeah. And so they don't go into just anaerobic respiration like that. Yeah. Okay, so the other two processes are called the citric acid cycle and the electron transport chain. Both of these actually occur inside of the mitochondria. Now, there's a lot of detail there. Let me take you to this slide right here. I would say that this slide is kind of what you need to know in terms of ATP production. Certainly you can, spend a little time reading through the details that I have on the other slides, but this is kind of the summary of what you need to know. There's 3 different ways that the cells produce ATP. The first step in the process is glycolysis, and it occurs out in the cytosol. Okay? Starts with 1 glucose molecule, and through that process you get 2 ATP and 2 pyruvate molecules. Okay? Now the 2 ATP you can use at any time. The 2 pyruvate molecules, if the cell is operating aerobically and there's not too much demand, it will take those 2 pyruvic acid molecules. Those will be moved into the mitochondrion, and then the citric acid cycle will actually use those pyruvic acid molecules as its source to then produce its own ATP and further the second step in that process. Okay? If your cells don't have the oxygen that they need, then the glycolysis and the production of the ATP through that way, that pyruvic acid actually ends up being converted to lactic acid, and that's what you hear about in terms of lactic acid buildup. Your muscles don't work, you got all this lactic acid building up because they don't have the oxygen to be able to allow that pyruvic acid to be used by the mitochondria instead. So the pyruvic acid kind of, in a way, decomposes into lactic acid, and that changes the whole pH of the cell, etc., and makes it not so good in terms of its performance. All right, but in the presence of oxygen, if the cell has oxygen, that pyruvic acid feeds into this. The citric acid cycle, which occurs inside of the mitochondria, cranks out another 2 ATP, and then it produces these molecules, NADH and FADH2. You don't have to know what those stand for, but it's basically, that's the molecular formula. You can see that there's hydrogen in there, right? See the H? Okay? These are hydrogen transporters, and what they're going to do is they're going to transport the hydrogen into this third step in the process called the electron transport chain. When it receives all those hydrogens, it's kind of like, have you ever seen those, I don't know, like in a river or something like that? They have those houses and they have a water wheel, right? And the water goes over the water wheel, turns the wheel, and produces electricity, right? The hydrogens that we get from the citric acid cycle, we use those, we kind of pile them up in one area of the mitochondria, and then they kind of trickle down, much like water going around the wheel. And through that movement of the hydrogen diffusing from one area to another, that gives the cell and the mitochondrion the energy to produce a whole bunch more of ATP. And if you look at the output there, you can see the electron transport chain puts out 32 ATP molecules. So now you're seeing a big payoff there. If the cell can get enough oxygen, we can go through these further 2 steps that occur in the mitochondria, and we can get a big, big payoff. Rather than just getting 2, we can ultimately get 32 ATP from that 1 glucose molecule originally. Okay? So that's the importance of the mitochondria. That's why they call them the powerhouse of the cell, etc., because their job is to do that, create lots of ATP so that it can be used in the cell to power what it needs to do. Yeah, so Jordan was talking about what we were talking about there. And Krebs cycle is the same thing as the citric acid cycle. Yeah. Once upon a time, there's a big movement in science where if you discovered something, you put your name on it, so then you're famous. And now they're kind of going the other way. They're going, okay, Krebs, it really doesn't tell us anything. And there's lots of different things we'll learn in the body that used to have like a name, like a bony prominence. It used to be called, somebody's name. And you're like, that doesn't tell us anything. Let's call it something that's anatomical, that tells us something about what it is or what it does. And so citric acid cycle has more to do with the biochemical process and the chemicals that are involved in it. And so it's a more anatomically correct name than the Krebs cycle. But they're the same thing. Okay? All right. So this slide here does a good job of bringing it together again. Like we said, this is outside the cell in the anaerobic reactions under anaerobic conditions. In aerobic conditions, that glycolysis that we talked about the pyruvic acid is used inside of the mitochondria to produce ultimately 34 ATP, 2 from the citric acid cycle, and then 32 from the electron transport chain. The other interesting thing that you see here is you look at this other product right here. What's that CO2 right there? Carbon dioxide, right? We have carbon dioxide being produced in the cells in this way. The cells need oxygen in order to produce ATP. At the end of this process, they are producing as a waste product carbon dioxide. The whole reason that we have a respiratory system is to support the cells in this way. They need oxygen, and they've got to get rid of carbon dioxide. So this whole system and the blood system and stuff like that is to help support them in their needs. Yeah? So lactic acid, that pyruvic acid that we said, which is a product of glycolysis, like we see here, if it's not used by the mitochondria, like we see there, then it chemically converts to lactic acid. And then it takes the cell much longer to convert that back to something that's not really toxic to it. So it's like a metabolic waste product that we have to get rid of. Yeah?
What prevents the pyruvic acid from becoming lactic acid? What can we do as
Well, on a very general level, that's why they say in terms of like aerobic activity is going to allow that whole bottom set of reactions to occur. So if there is oxygen present in the system, then the pyruvic acid is transported into the mitochondria and everything's good. It's only if there's so much demand on the particular cells, etc., that there isn't enough oxygen to do those chemical reactions, then it has to basically, if you will, decay that way and become lactic acid instead. So, in terms of if you think about muscles or something like that, so the more you train for long distance types of reactions or long distance types of activities, then the better your cells are able to get oxygen, your cardiovascular system works better, your cells are more efficient at using the oxygen and stuff like that, and so more chance that you're going to use the lower, more efficient pathway than have to resort to the pathway on the top. But, anytime you go over that threshold and you really increase the demand and there's not enough oxygen, it automatically reverts to the upper one. Yeah. Okay. So, where we are now is that we are kind of on our way to moving into our integumentary system. So we're on our way from going from cell up to skin. Now, like we said, there's the whole tissue level in between, but you guys are going to be addressing that in lab this week. So that may remind me, where are we in terms of our lecture schedule? What did it say for today? Did it say we're starting the integumentary system today?
Yeah, I think so. 6.
Okay. So what we'll find is that the skin and the integumentary system, even though on one level it is very complex where you're like, oh, darn, first we were just talking about cell, that seemed a little bit smaller at least. Now we're talking about a whole system. At least when we're getting into systems now, this is the easiest system in the body, okay? It doesn't have a lot of different components. It doesn't do a lot of complex physiological things. So it's kind of a nice stepwise process to get into the systems of the body and then work our way into it from there. So we'll do just a little intro of it today in our last 5 minutes or so, and then we'll work on finishing it up next time. But like I said, because it's not a very complex system, it shouldn't take us too long to get through it in its entirety. And then we'll be well set up for the lab this week and going over skin. Okay, so when we looked at the definition of a system is where we have more than one organ working together for a particular function. So like the digestive system, we've got like a pancreas and a liver, an intestine and stomach, and all these various organs that work together to help break down nutrients, etc. Okay? Now, below that, we've got an individual organ. And an organ, by definition, is where we have more than one tissue type making it up. And we said, like we talked about, like the heart as an organ. Okay? The skin is pretty odd in that it counts as both. So on one level, you hear about it like in terms of trivia, where it says this is the largest organ in the body, right? So it's the largest external organ. If you're talking about internal organs, it's more like liver or something based on mass. But it's the largest external organ in the body because when we get into it, we'll find that the skin is made up of more than one tissue type. It's a lot of epithelium, like we talked about stratified squamous epithelium out here. But when you get to the lower layers of the skin, like the dermis, then we're going to find there's connective tissue in there and stuff. And so, okay, it counts as an organ, more than one tissue type. But when we look at the skin overall, and we will, like in models and talk about it in lecture, when we look at all the details about things that we find in the skin, in fact, there are other organs, they're microscopic, but there are other organs that are inside of the skin. So, for instance, sweat glands, right? One of the overall functions of the skin is to help regulate body temperature, right? So you can get rid of heat or retain heat and stuff like that. So sweat glands help with that. Sweat glands is a lot of epithelial tissue, but there's like connective tissue holding it together and stuff like that. So we'll find that there are hair follicles, right? And the hair follicles, they actually have a blood supply going to them. There's nervous system receptors around them, so you can tell, like when the wind blows and blows your hair, you can actually feel and stuff like that. So because the skin has other individual organs in it, accessory organs, when we have more than one organ working together, then it counts as a system. So in that way, we call the skin the integumentary system, and it counts as a system of the body as well. Okay? The third classification of the skin that is a little bit interesting is that the skin also counts as a membrane. Okay? So a membrane is like a covering and lining. And so we talked about membranes before. Remember we were talking about, like, we had the parietal pleura, and we had the visceral pleura and the parietal pericardium, and the visceral pericardium, like on the organ and stuff like that. Okay? The skin lines us, so it counts as a membrane. This is the only dry membrane that we have in the body. The other ones are mucous membranes and stuff like that, but it counts as a membrane too. Okay? All right. So at least what we have is kind of the layout for how the skin is classified. And what we're going to do, really, we'll start next time with talking about different types of membranes and how the skin fits into that. And then we'll start looking at the skin in terms of an organ and the tissues that are involved, and then as a system and how it works to help you stay alive and maintain homeostasis. Okay? So that's where we'll pick up next time, and we'll finish up skin next time.